I did do some of this performance once before, except it was a whole lot easier. I teach in middle schools in Boston, and I got my students to do all the talking, and I got to just do the cool science demos. For this particular talk, the organizers raised the bar for me. They said, can you do what you did for the 6th graders, but talk about biotech at the same time? And I scratched my head, and whenever a challenge like that comes up, I see it as an opportunity. And the more I thought about it, the more I realized there is an opportunity here. And I hope by the end of this roughly half hour, I'll convince you that, in fact, some of the science demonstrations I do are somehow related to biotech. And when I was doing research on this talk, I found an author who writes for Wired, both the website and the magazine. He wrote a really cool book called What Technology Wants from Us. And he coined the term the Technium, the continuum of technology. Rather than saying that technology is something over there, it's really part of our lives, and we are, in fact, part of our technology. And I stretched that and thought of biotechnium, any way we manipulate biology. And one of the previous speakers in this room, and I won't give away the gist of his talk, talked about how biotechnology is really manipulating biology. And if you think about that, evolution has been manipulating biology for millions of years to propagate and further the DNA and keep that DNA code moving. We have been manipulating biology for tens of thousands of years. We domesticated plants and animals. That is a manipulation of biology for our purpose. And what I hope to convince you today is that that's what biotech is about. It's about manipulating biology, and it has a continuum that goes back tens of thousands of years, and potential, we have no clue how far it can take, because we are just now barely learning how to manipulate and modify biology. Let's see if that little red light is off. I believe it is, so give me a chance. The first way, and by the way, we're using and manipulating biology right now. You guys are using your brains to process what I'm telling you, but even before that, you guys are hearing what I'm saying. And the human ear is a fascinating piece of technology. It converts a physical phenomena, a compression wave, into some physical motion, the bones in the inner ear, and converts it to another sort of physics term, electrical energy, that our brain then processes and makes into voice or speech or the words that I'm saying. And this, of course, my 6th graders look at this and they go, it's a garbage can, Dr. Mantus. And I go, no, you got to think like scientists. This is clearly a vortex cannon. And I'm sure you all recognize it as such, and you all recognize it as a perfect model of the human ear. It's got a tympanum, and when the tympanum senses vibrations, it moves. But it's actually reverse of the human ear. What it allows us to do is it's going to allow me to show you what compression waves look like. The very compression waves that you use to detect my voice today. So I've filled it with some fog from a fog machine, and you'll see that you can visualize those compression waves using a vortex cannon. Our ears are amazing products of biotechnology. You mind taking this away, John? Thanks. So that's a vortex cannon. How is that related to biotechnology? Let me find a piece of technology I got to remember I have. This is a cochlear implant. It's implanted in patients' ears who are deaf. It's got some really interesting components. There's a processor that actually detects the sounds, recognizes them as speech, and converts them into electrical signals. The real meat and potatoes of this device, though, is this implanted portion. Right here, we have an array of microelectrodes that are implanted in the patient's, not in their brain, but in their inner ear. And instead of their own ear converting sound into electrical energy, this device does it for them. Those signals go to their brain. They don't recognize them as speech as we know it, but they recognize them as signal. And our brains are plastic enough, and I'll get to how good our brains are in a couple of demos, and you guys will have to help me with that, assuming you have very plastic brains. That's how they recognize speech, and they can convert these sounds into speech. So that's how we hear things. Let's think how we visualize things. Now, I'm going to talk about 3D visualization. This is, again, where physics and biology meet, and I think that is, by definition, biotechnology. 3D visualization. It's not going to be anywhere near as cool as Avatar, but I promise you it will be interesting. This is a 3D visualization of a microorganism found in the ocean. This was done in the 1800s. A scientist, German scientist named Haeckel, looked using nothing but a two-dimensional image from a transmission light microscope, the very inexpensive microscope sometimes they have in high school biology labs. By looking at that image, changing the focus, using his brain and his artistic ability, he was able to create a 3D visualization. I think we actually have lost something with modern technology because scientists don't have to be artists. In the 19th century and before, if you were a scientist, you had to be an artist. You had to think about how I'm going to visualize my data. Now today, it's easy. I'm no artist, but I can visualize data. Here are two bacteria imaged in with what's called laser confocal microscopy. You take lasers, not dissimilar from this laser beam, two different energies, image the cell. You can get three-dimensional information. You can get information about substructures here within the cell. It's the confluence of physics and biology. It is, in fact, in my world, I would say this is biotechnology. Here's two cancer cells imaged using laser confocal microscopy. We can actually go inside those cells with a laser, image particular substructures, but we can also destroy substructures. We can do micro surgery with a laser to understand how those substructures might affect the cell's behavior. So lasers provide us with an amazing tool in biotechnology. But not just on the research end. We can actually use them diagnostically for medicine, for humans. This is what's called confocal laser angiography. Those are blood vessels inside a living person's eye, imaged with light from a laser. You can tell it's an eye because you can see their eyelash. These are the blood vessels, and you can use that to look for abnormalities. Another way that physics and biology mix to allow us to diagnose disease and disorders. My next slide is actually, this is laser surgery. Laser surgery hurts? Laser surgery does hurt because it's a scalpel, and it's going to cut just like any other scalpel is. It might cut with a finer blade than a normal blade, but it's used to reduce bleeding. It's used actually more, and I don't mean to worry you, but used more in veterinary applications than in human applications. It's also, of course, used to correct vision. How do we convert that energy from a laser to do things like making a scalpel? Well, I'm going to use these balloons as models for cells, all right? And we're going to ablate these cells with a laser, and I'm going to demonstrate that. This laser pointer I'm using, you can tell it's a little different than a typical laser pointer. It's got a little bit more energy, and it's working at a slightly different wavelength. So I'm going to ablate these balloons by converting the light energy into heat energy. These are black balloons, so they absorb the energy. They absorb all the wavelengths of energy. They're going to absorb the green light. They're going to melt in a very tiny little hole. That melting of the latex membrane causes catastrophic failure. Some people call it a pop. I call it catastrophic failure. And I usually have my sixth graders count in their heads or count out loud how long it takes for a laser to ablate these model cells. If you can't see the balloons, you might want to move a little further over that way. And I think we have permission from the organizers. If you can't, you can move over there. All right, so I'm going to just count down 3, 2, 1. In your head, count how long it takes for my laser scalpel to ablate these cells. Now, what's interesting, it takes a little longer than you think. It's more because of the vibration than because of the lack of energy. It's not that the laser light has lost its energy when it gets here. It actually hasn't opened up much at all. But what happens, and you can think of that last poor balloon struggling for survival, it vibrates because the previous balloons vibrated. That's the challenge of laser microsurgery. You have to hold a scalpel that has such a fine tip, so carefully. It's almost impossible, or it is impossible, for a human to hold it without the vibrations coming into play. So that's an example of laser surgery on a balloon.